The overall goal of this procedure is to isolate and detect palmitoylated neuronal proteins from cultured neurons using a simple and adaptable biochemical assay. This is accomplished by extracting and immobilizing the target protein in the presence of N-ethylmaleimide, or NEM, to ensure the blocking of all free thiol groups. The second step is to treat the immobilized target protein with hydroxylamine, or HAM, to cleave the thioester linkage between the palmitoylated cysteine residue and the palmitate molecule. This frees up the palmitoylated cysteine's thiol group and makes it available for labeling. In the third step, the immobilized target protein is incubated with biotin BMCC, a thiol-specific molecule tagged with biotin. This biotinylates and tags the thiol group that was originally palmitoylated. The final step is to detect the level of palmitylation of the target protein by SDS page and Western blotting for streptavidin. Ultimately, the ABE assay is used to show levels of the lipid palmitate reversibly attached to a neuronal target protein, and can also detect subtle changes in target protein's palmitylation levels.
The main advantage of this technique over existing methods, like detection of protein palmitylation using metabolic labeling with tritiated palmitate, is that the acyl-biotin exchange assay is more sensitive and less time-consuming. The ABE assay also allows for more quantitative estimates of palmitylation and is optimally suited for detecting small changes in the levels of palmitylation.
Though this method can provide insight into identifying palmitoylated proteins in cultured hippocampal neurons, it can also be applied to other systems, such as primary neural cultures from other brain regions, heterologous cell lines, and even primary tissues.
To begin extraction of target protein from cultured primary hippocampal neurons, prepare the lysis buffer and NEM solution as described in the written procedure accompanying this video. Next, combine the lysis buffer and NEM solution by adding 0.5 milliliters of 2 molar NEM solution to a fresh 50 milliliter conical tube. Add 20 milliliters of lysis buffer over the top. Always add the NEM ethanol solution first and the lysis buffer second, so as to properly mix the two. In a 4 degrees Celsius cold room, quickly place the mixture on a rocking platform to fully mix for 5 minutes. Remove cultured primary rat hippocampal neurons from the incubator. Instructions on how to culture the neurons can be found in the text. After placing the cultured neurons on ice, gently remove the cellular media. Gently wash the neurons twice with cold phosphate-buffered saline. Next, add 300 microliters of lysis buffer plus 50 millimolar NEM to the first well of hippocampal neurons, and scrape cells using a disposable cell lifter. Collect the cell lysate and then discharge the lysate into the next well of that group. Scrape the cells within the second well using the cell lifter. Collect and repeat using a third well if needed to further concentrate the lysate. Collect the cell lysate into a pre-cooled 1.5 milliliter microcentrifuge tube. Add 100 microliters of lysis buffer plus 50 millimolar NEM to the first well and repeat the well scraping process to collect any remaining cell lysate. Pass the total cell lysate through a 26½ gauge syringe 5 to 6 times to aid in mechanical lysis, being careful not to blow bubbles into the solution. Nutate the cell lysate for at least 30 minutes at 4 degrees Celsius. Following centrifugation, collect the cleared cell lysate in a new, pre-cooled 1.5 milliliter tube on ice. Repeat the demonstrated harvest protocol for all groups of cells. Measure protein concentration in all lysate samples using the BCA assay as per manufacturer's instructions. Normalize the volume of all lysate samples from groups of cells to have exactly equal protein, with a recommended minimum of 500 micrograms. Top up all samples with lysis buffer plus 50 millimolar NEM to 500 microliters total volume. Next, add 1 to 5 micrograms of primary antibody directed against the target protein to each lysate sample. Nutate the antibody-lysate mixed samples overnight at 4 degrees Celsius. Before precipitating and immobilizing a target protein, prepare a 50% slurry of protein A or protein G coated saffrose beads as described in the written protocol. As a final step, add an equal volume of 50% slurry to each antibody lysate sample and nutate for at least 1 hour at 4 degrees Celsius.
In performing acyl biotin exchange, the proper pH and temperature of all the buffers and reagents, as well as their freshness, are critical to ensure a successful experiment.
Begin the ham cleavage with preparation of a number of tubes with lysis buffer of different pHs. The pH is very important for these steps and should always be adjusted using a pH meter. Per sample, prepare 2 milliliters of lysis buffer, pH 7.2, and 0.5 milliliters of stringent buffer prepared in lysis buffer. Also prepare 0.5 milliliters of lysis buffer plus 10 millimolar NEM per sample. Add PMSF and protease inhibitor tablets to all lysis buffers. Hydroxylamine, or HAM, is used to cleave the thioester linkage between the palmitoylated cysteine and the palmitate fatty acid. This is essential to free up the palmitoylated cysteine's thiol group and make it available for labeling with biotin. The omission of HAM cleavage can therefore serve as a useful negative control. Prepare additional 1.5 milliliter tubes on ice labeled as the -HAM control for each sample. Following the incubation of the antibody with the cell lysate, and then with the beads, gently centrifuge all sample beads at 0.5 times G for 1 minute at 4 degrees Celsius. With the tubes on ice, remove the supernatant and resuspend the beads in 600 microliters of lysis buffer plus 10 millimolar NEM. After resuspending all of the sample beads, immediately collect 200 microliters of the sample's lysate bead slurry and discharge into that sample's -HAM tube on ice, leaving 400 microliters of each sample's lysate bead slurry in the tube for the +HAM sample. This is to account for the limited degradation of the target protein caused by HAM treatment. 1/3 of the beads are used for the -HAM treatment, and the remaining 2/3 are used for the +HAM treatment. Add an additional 300 microliters of lysis buffer plus 10 millimolar NEM to the -HAM sample. Pipette an additional 100 microliters to the +HAM sample for a total of 500 microliters in each sample. Incubate the tubes for 10 minutes on ice and then proceed to wash the -HAM and +HAM samples as described in the written procedure. Following sample wash, add 0.5 milliliters per sample of lysis buffer, pH 7.2, to all -HAM samples. Add 0.5 milliliters per sample of HAM buffer to all +HAM samples. Then, nutate all samples for 1 hour at room temperature. Do not exceed 1 hour. Following HAM treatment, perform acyl biotin exchange by biotin BMCC labeling as discussed in the written procedure. Once biotin BMCC labeling is complete, gently wash the samples as described in the text. Remove the supernatant of the final wash, leaving the pelleted beads in a slurry with remaining buffer in the bottom of the tube. Dip a pipette with a small diameter tip into the very bottom of the tube through the slurry, and quickly collect any remaining buffer, being careful not to take up any beads. Add 40 to 50 microliters of 2x sample buffer plus 5 millimolar DTT to each sample. If necessary, vortex the beads to completely mix with the sample buffer, and immediately centrifuge at high speed to collect all beads in a pellet submerged in sample buffer. After boiling the samples and centrifuging again, add the complete volume of eluted protein in the supernatant from each sample to a single lane within a polyacrylamide gel suitable for Western blotting using SDS Page. Organize all samples so that the minus Ham control eluent and plus Ham eluent for a single sample are run immediately adjacent to one another during SDS Page. Following SDS Page, transfer to a PVDF or nitrocellulose membrane. Begin Western blotting with washing and blocking as described in the written procedure. Prepare an antibody solution of TBST plus 0.3% BSA. Add streptavidin HRP antibody, which has been reconstituted at 1 mg per milliliter in distilled water. Wash the membrane once in TBST for 10 minutes. Then, incubate the membrane in streptavidin antibody solution on a rocking platform for either 1 hour at room temperature or overnight at 4 degrees Celsius. Wash the membrane 3 times in TBST for 10 minutes on a rocking platform. In order to detect the palmitylation signal, expose the HRP luminescence using a chemiluminescent substrate kit. In order to normalize palmitylation levels to the amount of the protein of interest that was immunoprecipitated, strip the membrane using Western blot stripping buffer for 5 to 10 minutes on a rocking platform at room temperature. Repeat Western blotting steps using the primary antibody against the protein of interest that was originally used for the immunoprecipitation. Quantify palmitylation of the protein of interest using image analysis software and normalize palmitylation levels to the amount of immunoprecipitated protein. The ABE assay specifically detects thiol palmitylation of cysteine residues along substrate proteins and can be used to detect palmitylation of immunoprecipitated neuronal proteins. Treatment of the immunoprecipitated neuronal protein with Ham cleaves the thioester linkage between palmitate and a cysteine's thiol group, allowing for specific incorporation of biotin BMCC onto the newly available thiol group, which can be subsequently detected using Western blotting. The omission of Ham cleavage prevents the incorporation of biotin BMCC and functions as a negative control for specific palmitoyl biotinylation of the plus Ham sample. Therefore, the minus Ham control should always be run adjacent to the plus Ham sample for Western blotting by SDS Page. In an optimized experiment, the palmitylation signal of the neuronal protein delta-catenin can be readily detected by blotting for biotin BMCC at a concentration of 1 micromolar using streptavidin conjugated with HRP against parallel minus and plus Ham samples. The specific palmitylation signal for delta-catenin appears at its predicted size of 160 kilodaltons only in the plus Ham sample. The specificity of this signal was confirmed by reprobing for delta-catenin with the specific antibody that was initially used to immunoprecipitate it, resulting in a signal in both Ham treatments at the same predicted size. Optimization of the IPABE assay will result in a Western blotting profile of a minus Ham sample that is readily distinguishable from the plus Ham sample and exhibits minimal to no palmitylation signal. The concentration of biotin BMCC used to label palmitylated cysteines requires careful optimization, and if a supersaturating concentration of biotin BMCC is used during the ABE chemistry steps, excessive background and non-specific signals will be visible. The resulting streptavidin Western blot profiles among the minus and plus Ham samples for delta-catenin palmitylation appear similar when treated with 4 micromolar biotin BMCC, with additional background signals at different sizes, which indicates that inappropriate biotinylation occurred. Because hydroxylamine is a powerful reducing agent that results in limited degradation of the target protein, it is necessary to normalize the amount of immunoprecipitated protein used for minus and plus ham samples. Normalization requires the use of double the amount of immobilized target protein in the plus versus the minus ham sample, which results in indistinguishable Western blotting profiles for the target protein, as shown for delta-catenin. However, if the amount of immobilized target protein is not normalized between minus and plus ham samples, the resultant Western blot signal for the target protein in the plus ham sample can be lost, as shown for delta-catenin when equal amounts of protein were used in both ham treatments.
After watching this video, you should have a good understanding of how to extract your chosen neuronal protein from cultured hippocampal neurons, use ABE chemistry to detect if it is a substrate for palmitoylation in minimal time, and be able to adapt this protocol for use in other cell lines and tissues.